Welcome everybody, fantastic to have you online today. My name's Fern Hames, I'm from the Arthur Rylah Institute and I'm hosting you today. First of all, I'd love to, of course, acknowledge that we are meeting on traditional lands across the country today. Pay my respects to traditional owners and elders, past, present and emerging. I'm coming to you today from Taungurung country. So today is our 5th ARI Legacy Seminar. We're celebrating that ARI is 50 years old this year. On April the 8th we turn 50, so we're almost 50 and a half. And today is our 5th seminar. We've heard from Kim Lowe, we've heard from Matt White, we've heard from a series of women. We had Josephine, Tracy and Ethan Horlow talking to us about the future. And most recently we heard from Peter Mankhorst also talking about mammals. But today Lindy is going to pick up the story where Peter left off and talk to us about some of her history and some of the things that she's experienced in the more recent decades with marvellous, mysterious mammals. Thank you, Lindy.
Thanks so much, Fern, and thanks everybody for joining in and listening. Okay, so I hope you all heard Peter's seminar from a couple of weeks ago, where he did outline the research at ARI up until the mid-90s. And I'm following on from this, talking about specifically the mammal research at ARI since the mid-1990s. And in a lot of ways, I have actually followed on from Peter. He started his career working at the museum, and so did I, but a few years later. And then Peter was actually the person that employed me when I joined ARI way back in 1982. And unlike Peter, who was interested in wildlife from his childhood, for me, growing up on a farm, I was mainly interested in farm animals and didn't really get into knowing much about wildlife until after I went to uni, and then I joined the mammal survey group at the Field Nets Club. And I started going out on weekends, spotlighting for possums and trapping small mammals and going, wow, this is fantastic. And doing voluntary work firstly at the museum and then getting a job there. And then my first job at ARI was just for 3 months, and almost 40 years later, I'm still there. And I'll be eternally grateful to Peter, as I consider myself so lucky to have the dream job of having my work also being my passion, my hobby, and being at such a fantastic place to work. Now, I'm sure most of you are aware of what ARI is about, but we're DELWP's Biodiversity Research Institute, and because we're embedded within government, we can really target our research to address policy and management issues to inform decision making. And we've got a huge amount of expertise there to do robust, credible science that really makes a difference on the ground. And over the years, we've innovated a lot in the technologies that we use, and collaboration is a really big part of what we do, and so is teamwork. And most of what I'm going to talk about today has been a team effort, and I want to acknowledge all the staff who've contributed to these projects over the years. And down in the sidebar on the side, I'll mention some of the key ARI staff that have been involved in the projects, with just a few external really key people. So at ARI we study a wide range of both flora and fauna, but today I'm just going to focus on mammals. And we've worked on lots of different mammals, from feather-tailed gliders, kangaroos, smoky mouse, spot-tailed quolls, pygmy possums, brushies, dunnarts, long-footed potoroos, planigales, bandicoots, flying foxes, seals, whales, koalas, and also introduced species like deer and foxes and feral cats. And it's obviously not possible to cover all of that in one seminar, so what I'm going to focus on is just 2 main groups, the bats and then arboreal mammals. And both of these have challenges for studying. They're both nocturnal, they're both cryptic, often silent to our ears. You need lots of whiz-bang technology and ingenuity to study them, so they're certainly a challenge, both challenging groups to study, but hey, I've always liked a challenge. And so, as you can imagine, even on these two groups, ARI's done lots of projects, more than I can cover just in a single seminar. So today I'm giving a personal idiosyncratic account of some of the examples of our research over the years. And it's probably not going to surprise those of you that know me that I'm going to start talking about bats. I started working on bats in the mid-1970s, before much was really known about them in Victoria, and so everything we were finding out was new and exciting. They were these amazing little creatures that we knew so little about that I got hooked on them 40 years ago, and I still am. And because they're often so misunderstood, they've sometimes got a bad rep, but how could you resist a cheesy grin smiling face like this? This was the Gould's wattlebat, one of the very first photos I ever took back in about 1979, and I've got no idea what I was doing to it to make it smile so much, but I think it's just a lovely smiling face. And harp traps were invented back in the sort of late 70s, early 80s, and they were originally designed from US that got modified a lot by some people in Canberra. But then John Alderson, who was in our workshop at ARI, started making them, and between us we refined the design quite a lot to really get it to the amazing, well-designed and manufactured traps that they are today. And interestingly, because John was making them for me, he made them to the height, my height, so that I could put them up and down easily. And then I got a real kick out of the fact that when he retired from ARI and he thought he'd just have an easy retirement making a few harp traps for people, and because they were the only ones commercially made anywhere in the world, harp traps got sent all around the world, all to my height. So that was fun. So we started off doing broad-scale surveys for the Land Conservation Council, looking to see what fauna, and in my case what mammals, were in different areas. And so I got to start near Melbourne and go up to the northeast, the Murray Valley, East Gippsland, up in the Alps a bit, the Mallee, and then the Central Highlands. So I got to see a huge amount of Victoria in a lot more detail than I would have otherwise. And as Peter mentioned, this led to a massive increase in protection, going from, in 1970, less than 3% of the public land was in reserves, these red patches here, whereas by 2005, more than 40% of public land was in reserves. And while a lot of things contributed to this, having really good fauna data and being able to show where the important areas for fauna contributed to this. So we like to think that our surveys did have an impact in increasing the area of land protected. And during this time, we dramatically increased the number of records of bats. So this is lesser long-eared bat in 1970, when this is the database records for 1970. This is what it looks like now. So this isn't the species has increased that much, it's just our surveys, ours collectively, have increased the number of records considerably. So we've got a much better idea of where they occur. And for other species, the number of records haven't increased as dramatically, but we've got range extinctions. Eastern horseshoe bats were first found in central Victoria, and then we've expanded their range through that area. And we then started asking some more targeted questions, and working up in the Northern Plains and with Andrew Bennett and others, the use of remnant vegetation in farmland. And we were looking at a whole range of groups, but for me, for the bats, we really wanted to see how they were using the remnant vegetation, and was this different to other faunal groups. And Ross Meeks designed some of the earliest bat detectors that making these amazing contraptions that we started putting out in the field. And the way bat detectors work is picking up the high frequency echolocation call that the bats make and bringing it down to within our hearing range and to within something that we could record it on. And this is a slightly later version, when the detector themselves started being commercially made, but we were setting it up with a really old laptop and batteries out in the field. but I'll just play you a recording of the echolocation call. And so by having this, it meant that we could compare activity levels in different types of remnants, because trapping can often have some biases depending on how easy it is to trap the bats. So we could start looking at what was the relative use of different types of blocks of remnant vegetation. And we started looking at some of the, quite small patches of remnant vegetation and seeing just how many bats were out there and could use them. And this is an example up near Numurkah where we're working in this really highly cleared landscape and wanted to see what was in this largest patch of grey box remnant out in farmland. So we called into the landowner's house here and asked if we could go and try and catch some bats on his property, and he said, hey, I've lived on this property for 80 years, I've never once seen a bat, but if you're silly enough to want to go and try, you're welcome. Came back the next day, showed him the 50 individuals of 6 species that we caught that night, and he was just blown away by how many bats he had out there, and by showing that they consume large quantities of insects, including some that were quite bad agricultural pests, and how dependent they were on the big old trees he had on his property. It just hopefully gave him another reason to value and protect the remnant vegetation. And we started looking at even smaller patches of remnant vegetation, including scattered trees. And up till that stage, scattered trees were often thought to be really aesthetically valuable in farmland areas, but how important were they for biodiversity conservation? And so we included scattered trees in our sampling, and you might just be able to see a single harp trap between a couple of trees there. And we caught 29 individuals of 7 species in one night. And so they wouldn't be, all those individuals wouldn't just be living around those trees, they'd be foraging in the nearby remnant vegetation as well. But it shows just what the possibilities are if you've still got some big old trees in farmland. And another example is this 2-kilometre strip of roadside verge up near Pocola. And we caught 600 individuals, and we were marking them so we knew they were individuals, in a week in a 2-kilometre roadside verge. And that's far more individuals than you'd have for so many other groups. And it shows that the bats, if there's some remnant vegetation, particularly big old trees, they can hang on a lot longer than what some other faunal groups can do. And so then we moved on to asking even more detailed research on how are they using the landscape? What do they need to survive in the landscape? Not just where are they? And so we started looking at roosting and foraging habitat. What sort of trees were they roosting in? What sort of distances were they moving to forage? And we did some early radio tracking with these tiny transmitters that only weigh about 0.4 of a gram that we attached to the bats. And by following the transmitter around, we could then be able to look at the sorts of trees that they were roosting in. And these bats confounded us by moving much greater distances than anybody had expected before, because lesser long-eared bats are thought to only move small distances between roost site and foraging area. And then we found that they were moving up to 13 kilometres between where they were foraging and where they were roosting, with a lot of the roosts up in the Barmah forest. And so to do that, we often needed to get a plane up to be able to radio track from the air. And it was really lucky to have John Sillins working with me, who was just such a technological whiz. He managed to make everything work, and we got some fantastic data from it. So we started being able to see what type of trees they were using. So Gould's wattlebats was typically roosting in big old live red gums in this case, but they can use other species, and often a dead spout on a live tree that about 20 or 30 individuals would roost in there and fly out. Whereas lesser long-eareds were tending to roost in big old dead trees. and often trees that had narrow cracks on the main trunk. This 2.5 centimetre crack here had 36 females and their young fly out of there one night. So when I'm talking to landholders about the trees on their property, I try and explain that not just the really big hollows are important, they're important for some faunal groups, but even a narrow crack on a dead tree can be a really, really important roost site, and it gives people an extra reason to protect those trees. So they were two common species, and they were being quite specific in what they were doing roosting wise, but what about some of the rarer species? So the southeastern long-eared bat is listed as a vulnerable species, and very few records for Victoria. Prior to when, during our surveys we found a couple, but then prior to the more substantial study that we did in the late 2000s, there'd been only 6 records for Victoria, despite a lot of survey effort. So we wanted to try and work out why were they so rare and why were they so restricted. And where we found them was in these areas of really old growth mallee, and up in the Sunset Country there've been such extensive fires through those areas, big fire scars, that this patch of old growth mallee here is the only place that we found the species to occur in. So this is the Nowinji block in the western part of Hattakolkine National Park. And I think it's because this old growth mallee, of which there's so little left of in Victoria, and it's potential that there's been a bit of the Rark Plains, the saline plains, may be a natural fire barrier to keep this area, because it probably hasn't been burnt for a couple of 100 years. And so working out how susceptible this species is to fire, we've been able to provide that information so it can be recognised in fire planning, both in terms of preventing there being planned burns through there, but also trying to stop wildfires going in there. So, but we've still got lots of unanswered questions as to why they're so rare, but they seem like they're still hanging in there. It can be really frustrating when you can't answer the questions as to why a species is so rare and why they are declining, and sometimes this can have dire consequences. And the Christmas Island Pipistrelle is an example of this, and I've had the dubious honour of having the longest association with this species from first identifying that it was declining and being there on the day it went extinct. And so this is really tough and quite emotional. So I first visited Christmas Island on holidays. Richard Hill was working over there on owls, and so I went over to visit, and I thought, well, I'll take a harp trap with me and a bat detector, as one does when they go on holidays. And so I went over to the island, and the island of course is famous for the red crabs, which are so impressive. But the little bat was much more poorly known. It was the only microbat on the island and was endemic to Christmas Island, so only found on the island. And it's a tiny bat, it's only 3 grams. And so when we were there in '94, it seemed like they weren't as common and widespread as has been recorded in the only other study that was in the 1980s. And so I raised the concern to say, I think there might be something happening with this species. So we managed to get some funding to do a proper trip there, and this was an ARI trip to Christmas Island. So our work isn't just in Victoria, we do work in other places as well. And each time we managed to get some funding and get there, the species had declined further and was shrinking further west. So they used to be widespread all over the island, and by '98 they disappeared from most of the eastern part of the island and were shrinking west. They were only commonly found in the west of the island, and then each time we went they were shrinking further and further west till they only occurred in this far western bit. And by using bat detectors at standard sites, we could quantify what the decline looked like. And over the years, this is what it looked like, which became devastatingly obvious where that trend line was going. And so we tried investigating roosting and foraging and activity patterns and diet and threats, but we could never work out, even though there were lots of potential threats, we could never work out exactly why the species was declining. But I always thought it was the wolf snake, and that seems to be the consensus now. So we desperately tried to convince everybody that we needed to be able to set up a captive breeding colony, which would require a rescue attempt. And I was there in January in 2009 and could only find 14 individuals left, and predicted that by June the species would be extinct. But despite all my pleading, we were only granted permission to go in July and got there in early August, and it soon became apparent that there was only one individual left. So in reality, we were already too late, but we desperately tried to catch this last individual. We had a large number of detectors set out each night so we could actually follow where this one individual was going, the general area it was roosting, the general area it was foraging. Then we tried everything to try and catch it. We had harp traps out everywhere. I designed this crazy mist-net funnel trap that we built over there to try and catch them. The last known roost, which they were using in January under these flimsy bits of bark. Ross Meggs designed this amazing freestanding ladder, and we designed this contraption that we could try and wrap around it, but they weren't using that roost when we got there in August. And so we'd been monitoring this bat every night for a couple of weeks, and then one night it went silent as well. And I knew that that was the day that the species had gone extinct. And for a conservation ecologist, there's no greater failure than not being able to stop a species going extinct. And I was devastated by it then, and I'm still devastated by it now. But rather than giving up, it made me more determined than ever to do whatever I could to stop other species going extinct. And so I've kept on working on critically endangered species, and now doing a lot of work on the southern bent-winged bat. And it's a species that occurs just in southwest Victoria and southeastern South Australia, and there's only 3 maternity sites for it, and it's declined pretty dramatically in numbers over the last 4 or 5 decades. And so a couple of years ago I wrote the recovery plan to try and guide the recovery actions, and we're doing a lot more work on this species to really try and see what's happening and try and prevent its decline. And it's really important to get accurate numbers as part of any monitoring program, to both look at the trends and also to assess the effectiveness of management interventions. However, when you've got up to 20,000 individuals flying out of a cave in the pitch dark, it's quite hard to count. And while it might sound like 20,000 individuals is a lot, because the numbers have declined hugely, that's why the species is called, classified as critically endangered. But 20,000 individuals is pretty tricky, so we need some flash technology. So a few years ago, started using a thermal camera with some missile tracking software that was designed by the Army in North America. And what it does is follow, in complete darkness, follow the trace of the bats using the heat signature. So in this one, the bat is red with the blue being just the trail of the heat. And this is over at Narracourt, and it seems to work reasonably well over there, but the main maternity site we've got in Victoria at Warrnambool, with the different size and configuration of the cave mouth, made it really difficult to use. And plus it was based on really old hardware and software, which became really clunky. So Amanda and who works with me on this, and her wonderful husband Pete, decided they'd design a new version of it called Bat Tracker. And so it's doing the same thing, but in a much higher resolution. It's not as colourful, but you can see much higher resolution image, and so we're picking up a much greater number of the bats that are coming out. And being able to calibrate it, we can really fine-tune it to get the actual numbers, and also incorporate uncertainty levels. So it's made a really big difference to be able to get good estimates of population numbers, and especially now that we also monitor the two nearby non-breeding caves that we know the bats interchange between a lot. So we're now getting some really good baseline data that we can start to measure trends in the future. But we also need to get a good idea of the number of pups that are born, so the number of young. And to do this in the cave, and this cave is massive, it's 40 metres high inside, so we use the thermal imaging camera to look up, and this image up here is all these little dots are bats, with a big cluster of bats and some flying ones there. And just, it's pretty like, amazing little fireflies flying around, watching the bats flying around. And here, just panning down to where some of the adults were roosting up the top, to where this big cluster of young, where the young are born in the cave. So again, we've got to do this in the pitch dark because any light disturbs the bats. But then the next challenge is trying to count those. And so this is, it's a bit dark, but this is all done at about 25 to 30 metres away with a 400mm lens on a camera that's been modified for infrared, and with some really bright infrared illuminators. And when they're tiny, this is all the young through here, and the adults flying in and out. So all these tiny little jelly babies are the newborn young, and trying to count all of those to get an accurate number of those, so we can start looking at things like breeding success. So while we're getting a good handle on numbers at the main breeding caves, we still don't really know the full extent of the range. And at the moment, southern bentwings are listed as, or defined as a subspecies rather than a full species, but that might change. It occurs in southwest Victoria, and the very similar looking eastern bentwing bat is in eastern Victoria, with an overlap in the eastern Otways. But we didn't really know to what extent, how far the range went. So an area of mines, because all the through central Victoria, where there aren't any natural caves, what the bats are roosting in there are mines, so disused horizontal mines. And so we managed to get a project of searching lots of mines, and I love going around, crawling around in mines, so that was heaven for me. And managed to, with all the right OH&S requirements, managed to find bats in as many of those as we could, and caught them and took genetic samples, because at the moment the only reliable way of telling them apart is on genetics. And found that all of them were eastern bentwing bats and no southerns. So it's helping to delineate the range, but we still need to do more work to be able to get a full understanding. But it also highlighted that some of these mines that have been around for 150 years or so might need some active management to keep them open as key bat roosts. So what else could be causing the decline with southern bentwing bats? We think that a lot of it is around habitat issues, but we wondered whether health issues could be part of it as well. And so, but the health and the veterinary side of things is outside of my expertise. So a collaborated with Peter Holtz, who's a really experienced wildlife vet who was keen to do a PhD on something interesting, so talked him into doing it on southern bent-winged bats, which was absolutely fantastic. And so Peter and Jazzy's other supervisor, we looked at a whole range of viruses, fungi, parasites, internal and external, pathology, haematology, to see if there was anything that might have been the cause for the dramatic decline. And while there were some health issues and some interesting patterns, there didn't seem like there was any one major thing that might have caused the decline. But it's given us some fantastic baseline information now that we can compare in the future. But could there be some emerging threats that aren't yet here? And the key one is white nose syndrome. And it's a fungal disease that occurs naturally in Europe, but was introduced to North America. And since 2006, it's killed over 6 million bats. So it's having a massive impact in North America. And we're really concerned that if it accidentally got introduced into Australia, it could have a similar really dramatic impact here. And from Peter's work, we think that it's probably not yet in Australia, but we did a risk assessment to see what the likelihood it was of being introduced and what the most likely pathway would be and what impact it would have. And the species that we thought would have the most impact on was the southern bent-winged bat, unfortunately. So it's something that we want to do a lot more work on, and desperately working with quarantine people to see if we can, and other groups that might risk bringing it in, to see if we can keep it out of Australia. And this was an example of a project that just wouldn't have been possible without the collaboration. And often the collaborations includes working with students, and I love working with students because it's a great way to investigate lots of other questions that I wouldn't be able to do. And so in addition to Peter's work, I've been fortunate to co-supervise a whole range of PhD students. Emmy's currently working on doing amazing stuff with population dynamics of southern bentwings. Amanda, in the other half of her time, is looking at a lot more detail on southern bentwings, where they're roosting activity, where they're foraging. Feely's looking at landscape configuration in bats and developing some new technology for looking at echolocation calls. Steve's been looking at the efficacy of nest boxes as a conservation tool for bats. Manisha was looking at the impact of roads on bats. Tanya, the role of wetlands in urban areas. Lisa looking at roosting behaviour and the influence of parasite load. Susan on large-footed myotis. And then a whole lot of honours and master's students that I've been fortunate enough to co-supervise over the years. And I get a huge amount out of this because they're things that I wouldn't necessarily be able to do myself. They're great questions, really great students. I really enjoy the mentoring, both in terms of the questions and the working out what to do, but also helping out a lot with fieldwork, because I get to go out and do lots of fieldwork, and it's lots of fun. So I get lots out of it, as well as hopefully contributing to their projects. And I also get lots of fun out of giving presentations to community groups. Trying to change attitudes is one of the reasons why I'm so passionate about working on bats. And people are often surprised just how small the bats are. And this is a fully grown little forest bat sitting on my thumb that weighs just 4 grams. And showing people the huge amounts of insects that they consume. So this is a lesser long-eared bat feeding on a mealworm, but these bats can eat up to half to 3/4 of their own body weight in insects, and so they're really important at consuming insects. And I always get a real kick out of when I'm giving a community group talk and somebody comes up that says they've been to one of my talks 15 years ago and they remembered the talk and the dead tree in their back paddock that they were going to cut down for firewood, they've left up there for the bats, and so it makes all the difference. And talking about making a difference, I'll now move to the second half of this story, which is about research on mammals within the timber harvesting space. And timber harvesting is an issue that polarises the, contentious issue that polarises the community. And what we aim to do at ARI is to do good quality science so that the decisions can be based on evidence, the best available evidence. And so our role is to collect the data, analyse it, present it in a way that it can be clearly understood and the implications of different decisions can be fully explained. And we've been working in this space since the 1970s under various projects such as the silvicultural systems project, the early pre-logging surveys and the early RFA surveys. But I wasn't involved in any of those, and I didn't start working in this space until 2011 on the Forest Biodiversity Project. And the aim of this project was to improve our understanding of some key threatened species to enable the management to move away from a detection by detection approach to more a landscape scale approach, so that if we knew the most important areas for each of these species, then the management and protection could be targeted at those. So we did surveys for a whole range of species, Longford potteroo, smoky mouse, large forest owls and gliders, some aquatic species and Leadbeater's possums. And we needed a survey technique that we could survey Leadbeater's possums rapidly at a broad scale. And so we linked up using the thermal cameras that I was talking about before with using the call playback technique that people like Dan Harley and David Lindemeyer had used successfully before. And I'll just play you a bit of the call that's a social call that can use that can really attract the animals in. And so using that call in conjunction with being able to pick up a little light that we could then get a spotlight on to be able to work out exactly what the species was, we could survey a large number of sites across the whole of the species range and then start doing some occupancy modelling to be able to work out where the areas that they were most likely to be occurring in now. And one of the things this showed up was the huge impact of the 2009 fires, which burnt about a third of the species range, where it seemed like they disappeared from during the fires. But we also want to understand how the population might change over time. And so we did a PVA that was led by Charles Todd, which modelled the future trends based on past fires and predicted changes changes in the future to the forest structure. And what it showed was a marked decline in 2009, where they dropped, but also this continuing decline, which is based on the big old stags that were killed in the 1939 fires, falling over time. And this was exacerbated by modelling in future fires as well, that was showing we're going to get this real bottleneck over the next 50 years or so, where the habitat would become less suitable. And so this and data by other groups working on leadbeaters showing a decline in the species contributed to the establishment of the Leadbeater's Possum Advisory Group. And this was a group that was appointed by the Minister to make decisions using a consensus approach around things that could be done to support the recovery of Leadbeater's possums, but while maintaining a sustainable timber industry. So it had those dual objectives. And we wanted to use the best available science for this. So my role with LPAG was to provide the science, and not just what we did at ARI, but all of the information on Leadbeater's possum, to present it to the advisory group in such a way that they could weigh up the cost effectiveness of a whole range of potential actions to see what would be the most cost effective within the terms of reference. And so the group came up with 13 broad recommendations, and because they'd agreed with it within the group, all of them got accepted by government and all of them have now been implemented. And one of the main recommendations was to establish 200-metre radius timber harvesting exclusion zone around new and existing colonies. But so we needed to be able to find as many colonies as possible to be able to protect. And so our job was to rapidly locate more colonies. And so we came up with another technique for doing this, using the heat and motion cameras that had been used on the ground and also low down in trees for leadbeaters. But we coupled that with using arborists who could put the cameras at whatever height we thought the animals were using, sometimes up to 50 metres above the ground. Mostly we were recording the animals at 25 metres. But it was really useful to get them to that height where the animals were moving. So we had a camera set up, and then the bait, which was the lure of creamed honey that the animals loved. And so the first 2 years of our targeted surveys, we really focused on areas most likely to have leadbeaters, because the whole aim was to find as many as possible, as quickly as possible. So we sampled almost 300 sites and found leadbeaters at 52% of those sites, with a detection probability of greater than 85%. So that's if the species is there, it's 85% likely that we'll actually detect them, which for any survey technique is really, really good. And so it meant that wherever they were found, they'd get these 200-metre radius exclusion zones around it. And of course, having the photos, we get all sorts of really cool photos like this, where a leadbeaters is coming up towards the bait and a mountain brush-tailed possum, the bowbuck, is sitting there going, whoa, this is mine, and then looking like it's about to pounce on it, and then they actually touch noses underneath. So it's fun being able to look through all the photos as well. Now, that was really useful to find as many colonies as quickly as possible, but it didn't enable us to extrapolate across the range because we were just targeting the spots that we thought were most likely to get them. So in the third year of the surveys, we selected our sites randomly to be able to sample right across the range of the species, and so we could extrapolate. And here we recorded them at 37% of sites, which I found really encouraging in that it was at a higher rate than what we might have previously thought. But despite this, as indicated earlier, there's still going to be a bottleneck in hollow availability over the next 50 years. So another project was whether we could design construct a hollow that the animals might use. And what they like is a natural hollow with a small entrance dimension, but with a great big huge cavity inside. So working with Mark Cashmore from Tree Tech, designed a cavity that they could put in with a chainsaw, and arborists are always bright chirpy people, and so constructed this big internal cavity and with just this small entrance dimensions. And it's been really successful. We've got leadbeaters using, we've been monitoring them for the last 5 years now. We've got leadbeaters using almost 60% of them, building their big shredded bark nest and using them throughout the year and for breeding. This is a female with 2 young on her back, and I'm not sure how they're all going to squeeze through that hole with the young not getting knocked off, but they seem like they manage it. And we can get large numbers all in the one hollow. This is an example of 5 individuals coming out of this hollow over a 4-minute period. And so this has been able to show, this trial has been a really useful, showing it's a really useful technique for supplementing natural hollows where they're in short supply. Now, we obviously still need to protect natural hollows wherever we can, but this is useful for supplementing. Now, another forest-dwelling species that's currently in decline is the greater glider, and it used to be a common species when I started doing surveys back in the 1980s, but it's now so much rarer. And we really need to understand where it's occurring now, because the records from the 1980s and 90s aren't as relevant now because we don't know if they're still in those areas. So we really need to update our distributional knowledge. And while they're big and they look pretty obvious, sometimes you don't see greater gliders every time you go spotlighting. So we wanted to be able to actually quantify abundance and density estimates, not just the numbers seen. So developed a mark recapture distance sampling method where two observers go along a transect 10 minutes, one behind the other. And then by being able to assess which individuals were seen by which observer can actually work out densities. And this has been a really good technique that's now being used as a standard approach in other programs like the new Forest Protection Survey programs. And surveys in the Strathbogies showed that there's still some really good densities of greater gliders in those areas, but in other areas it seems like they're declining massively. And so some of this information helped to contribute towards options for the action statement. And when the action statement came out, that was when the Minister announced in November 2019 that timber harvesting would be phased out from native forests by 2030. And so it was a fantastic day, and I was really lucky to be able to be up there with the Minister in the Strathbogies with the local environment group when she was making the announcement. And so it just shows that, you know, lots of other information went into this and lots of other efforts, but you know, our research can make an impact. And all of these things have led to 2 awards that I've been given that have humbled me hugely. The first was the Northern Freetailed Bat being named after me, and as a bat ecologist, there's no better thing in the world than having a bat named after you. So that was huge. And then being given the Public Service Medal last year, and it was fantastic to be able to share this with James Todd and Kim Lowe as well, and to see biodiversity conservation being recognised in the awards. And I see that this award was not just for me, but for all of ARI, as I believe that it was the work of the whole team that was being recognised. I was the one up there getting the medal, but it was all of ARI, not just me. And none of this would have been possible without ARI. I've been so fortunate to be part of this great institute for so many years. It's a fantastic place to work with great people that are passionate, doing research that matters and having lots of fun. And I look forward to the next 50 years of fantastic research out of ARI that makes a real difference for biodiversity conservation. And I'll finish it there, and back to you, Fern.
Oh, Lindy, bravo. I can hear everybody around the state applauding as well. Thank you so much for that really wonderful journey through the last few decades. It's a privilege to have heard from you about that today. I just love that story. It's so good. I mean, you've really reminded us of the critical nature of habitat, of the excitement of innovation in technology and what that allows us to do. And of course, I loved hearing from you about the many staff, students and landholders that have been inspired by you and continue to be in your story about science that matters, science that makes a difference. So bravo and thank you so much. Andy is going to help us with some questions in a moment. There are some of the questions you'll go, yep, we always get that question. So there's a few usual questions there and some other things as well. But while Andy's just having a quick look at those, I just want to remind other people of two things. One is you can see on the slide there that our next one coming up will get out of the trees and into the rivers. We'll be looking at John Kewen telling us about the aquatic story for ARI. So come along for that one on the 19th of October. And if you've missed any of these, or you know other people that would like to watch these, the recordings for all of them are on our website, on the site that's listed right there. So please jump on and check those out if you'd like to see them again, or you know others who might like to see them, or you've missed them. So Andy, help us with some questions that Lindy can help us with the audience.
Yeah, no worries. We've got some pretty great ones, and I'm going to start with a question I had once upon a time myself. How do the southern bedwing bat mums locate their own babies? Is it by scent?
Fantastic question. I'd love to know exactly the answer. They probably know where they've left their young, and then by scent and also by sound. There were some experiments done in North America in a colony of 2 million bats, and they thought that there was no way that individual females could find their own young, and so they marked mother-young pairs, and sure enough, even in a cluster of 2 million bats, the mother came back and found her own individual young. So it's pretty amazing. The bond between mother and young is really strong.
It's incredible. I love that. It's so good. We've got a couple of questions around nest boxes, so I'm going to group them together. But these are questions around bats. How can we create habitat for bat boxes in urban wetlands? And is there any way we can sort of protect them from cats and things like that in urban areas?
Okay, so bat boxes is something that we've been doing a lot of work on, and Steve Griffiths, been working with him, and he's looked at them a lot. And we know that bat boxes do get used a lot by bats. It's largely a couple of the common species. It doesn't seem like it's all species will use them, and it's often Gould's wattlebats that will be the key ones that will use them, and particularly in Melbourne in urban areas. But it can be really useful as an adjunct because what the bats need is multiple tree hollows to move around between. And so if they're getting a bit short on natural tree hollows, by having a few extra nest boxes can really help. But it can't be seen as a silver bullet for meaning that you don't need to protect big old trees with hollows, because it's not the whole faunal, the whole bat community, not all species can use them. But certainly they can be useful, and there's quite a few different designs around, and you can buy them commercially. The key thing is to have them up reasonably high and with an opening underneath, an open space underneath, so that bats can sort of drop out and fly down, and no little platforms or ledges that something like a cat could sit on nearby and try and grab them. So just on a tree trunk or the side of a house or something, 4 or 5, 6 metres up with open space underneath, and they'll probably come and use them.
And regarding, I guess, your work with community, there's a question around your involvement with bats and the Indigenous community. Do you have any experiences or programs that you'd like to shed light on?
We don't have a huge amount. I'd be really fascinated to hear whether there's some bat species that are totem species, or there's this really interesting story from back in the mid 1850s at an expedition that went up to the junction of the Murray and Darling, and the Europeans that went up there and had a lot of the local Indigenous people helping them, catch all the animals. And there was a comment saying that they wouldn't bring in bats because bats were seen as departed friends and relatives, and so they wouldn't collect them. But we don't know a huge amount otherwise, and I'd love to find out more if anybody's got any information.
Great. A common question, I guess, around bats again. do any of the Australian bats harbour viruses like some of their Asian cousins?
That's a whole huge topic. So there's all animals have viruses. The PhD study that I mentioned, Peter looked at and found a few viruses in southern bentwing bats. All bats have some viruses. It's whether those viruses cross over to other species and cross over to humans. So there's the Australian bat lyssavirus, which is the rabies-like virus, and it is something that can cross over to humans. And so for it, the only way it can be transmitted is the bite of an infected animal, and it's incredibly rare in the bats, but the bite, so the saliva going into an open wound. So if you don't get bitten by a bat, there's no risk whatsoever. And so we wear gloves when we're handling them, even though most times the bats don't bite because they're very gentle, placid animals. But if anybody is picking up a bat that got caught in a house or something like that, just make sure you've got something over your hand, and then there's no risk from that. The whole coronavirus thing is a much bigger story. There's no evidence of there being coronavirus risks for bats or for people in Australia, but it's a huge topic that we don't have time to go into here.
Thanks very much for the answer. Regarding citizen science, what is or what could be the way people can contribute to ARI science, and what kind of roles do you see this having in future?
Yeah, citizen science, there's a few good examples with that. Some of the things, because we need so much technology, some of the some things aren't easy to do. But with the flying foxes, and I've talked all about microbats rather than flying foxes today, but there's regular flying fox counts of the Melbourne colony where Rodney Vandery leads that and has people every month, outside of COVID times, every month to count the flying foxes coming out, and that's been a fantastic program and needs lots of people. And so that's one thing. With the bat boxes, we've got bat boxes out at Organ Pipes National Park and a few other places in Melbourne, and normally we check those every couple of months and people can come along and see the bats doing that as well, which is great. In terms of the microbats, people getting their own bat detector and recording. Some of the community groups are doing that really well. It takes a bit of effort because you need to be able to identify the calls, but that's a possibility as well.
All right, I know we're a little bit over time, but I've got one last question, I think, before I throw it back to Fern. This is regarding Leadbeater's possums and the connectivity between groups. In terms of their management, should we be increasing connectivity and not isolating them, or is there something else that makes it a bit more complicated in the way we protect that species?
So connectivity as in, so leadbeaters need lots of connectivity within their home range to be able to move through it. So they like really dense vegetation to move through, and then there's connectivity between groups as well. So having connectivity over roads, for example, is important to enable animals to move around. So dense vegetation is really important, but also keeping it connected at that broader scale is important as well.
Awesome. Thank you so much, Lindy. I think we're going to have to close it up now. Fern, I might throw it back to you.
Yes, I think so, Andy. I'm sorry, everybody, we didn't get to all of the questions because there are so many, as you might imagine. So, and sorry about my dog in the background. Timing is everything, hey? Thank you, everybody, for coming along. Thank you, of course, to Lindy. Wonderful talk, as always. If you've still got questions and you want to know more about Lindy's work or bats or arboreal mammals, go and have a look at the ARI website, and there'll be lots of her publications and work in there. And we hope to see you at the next one when Jono comes and talks to us about all the fishes. Thanks very much, everybody. Thanks, Lindy. Bye-bye.
Bye, everybody. Thanks for listening.